Thank you for having me here at ISMB 2018, and I hope everyone is enjoying the wonderful conference and the beautiful city as well. Today I will talk about how genome editing, in particular CRISPR, intersects with machine learning, and of course how we have helped advance the state of the art in that respect. This is joint work with Professor Hyungbum Kim's group at Yonsei University, and based on the recent paper published in Nature Biotechnology. I am one of the two first authors. The content of this presentation are as follows. To begin with, I will just give you a brief summary of this work first, and then I will explain the background, proposed methodology, experimental results, and so on. So the problem we are interested here is which DNA target will be cut more easily with the CRISPR-Cas9. This is often called on-target activity prediction. There will be multiple targets available, so we want to predict CRISPR activity of each target, then cut those with higher efficiencies. There are some computational approaches with rule-based or some machine learning algorithms. However, since they require manual feature engineering and have difficulties in dealing with the data sets under different factors, they usually have shown some limited performances. The heat map below shows some evaluation results of previous approaches, where the rows are data sets and the columns are the prediction approaches. As you can see, most of them perform well on the training data set, which are indicated by the hatchet boxes right here. But they mostly fail on the other data sets. Therefore, we present our solution. First, our collaborators generated large data sets by high-throughput profiling of CRISPR-SHPF1 activities. Then, our group developed a deep learning-based regression model, which we call DeepSHPF1. In this presentation, I will focus on the second part. DeepSHPF1 is based on a convolutional neural network, has a multimodal architecture for considering different factors, and trained with pre-training and fine-tuning strategy. Now let's move on to more background. CRISPR is an innate adaptable immune mechanism of bacteria, of which now we take advantage for the efficient targeted genome editing. By using the artificially engineered nucleases, we can now delete, insert, or replace DNAs in the genome. By the way, there are like two ways that people use the term genome editing. The first one is like replacing a nucleotide like A to T. Then the other is gene knockout. Remember, for this presentation, I will use the term as in the gene knockout. So we just want to stop a gene from functioning by cutting it with CRISPR. There are two main components for the CRISPR technology. The first one is an endonuclease, which actually cuts the target sequences. And the second one is a guide RNA, which directs the endonuclease to the target sequences. However, the CRISPR complex cannot actually cut all the target sequences. CRISPR has one constraint where it can be deployed. That is, it can only cut the target sequences next to a short motif called PAM, protospacer adjacent motif. This is one of the reasons why researchers have searched for different endonucleases having distinct PAM sequence. Currently, the most widely used ones are Cas9 and SHPF1. In this work, we focus on the SHPF1, which has the TTTV PAM sequence. Now, the problem here is that we don't actually know which target will work well. Some of them will do a great job, but some of them will not work at all. In reality, you will have thousands of potential candidates for a given gene on where to cut it. So you cannot try every target sequence for every time you want to knock down a gene. This is where we use machine learning. Using the large dataset my collaborators generated, we built a predictive regression model. We call the deep learning-based CRISPR-SHPF1 activity prediction model as DeepSHPF1. Deep learning is a branch of machine learning algorithms which use neural networks with multiple nonlinear layers. The conventional machine learning algorithms normally have two-step procedures. First we perform feature engineering, then train some classifiers with the handcrafted features. On the contrary, deep learning can relieve that laborious manual feature engineering by exploring the neural networks. We can learn hierarchical features solely from data as a representation learning method. For example, in image, the deep learning algorithms can learn edges in low levels, parts in mid levels, and finally recognize the objects in the higher levels. Among a lot of different deep learning architectures, in this work we use a convolutional neural network. CNNs have great advantages in analyzing spatial information, considering the data locality. In this work, we use CNNs. They have been widely applied in various domains and grid structure data, such as images and sequences of words or DNAs. For the following, I will explain the proposed methodology. We don't entirely know what determines the CRISPR activities, but there are two critical genetic and epigenetic factors. The genetic factor is a target sequence composition, which dictates their thermodynamics. There were a few options that we can use for the length of the target sequence, and we found that 34 base pairs were adequate by cross-validation. It includes 4 base pairs upstream of the PAM sequence, 4 base pairs for the PAM sequence, 20 base pairs for the protospacer, which hybridizes with the guide RNA, and 6 base pairs for downstream of the PAM sequence. I mean protospacer sequence. And the epigenetic factor here is chromatin accessibility, which we call CA here. The same target sequences can have different CRISPR activities based on their accessibilities in the genome. Generally, if the target is more accessible, it shows higher activity because the CRISPR complex can approach the target more easily. In this work, we use DNH1 hypersensitivity as an indicator of chromatin accessibility. It is provided by the ENCODE consortium and indicate the availability of DNA to DNH1 enzyme. So given considering the two factors, what machine learning engineers would want to do is like this. We want to use a data set which reflects both factors, the genetic and the epigenetic factors, and train a model that can predict the CRISPR activities. However, in reality, we do not have enough data of this kind. Instead, we have a larger data set which reflects only the genetic factor, in this case, the target sequence position. This is called the integrated target data set, obtained from high throughput but somewhat restricted experiments. And the former one is called endogenous target dataset, obtained from low throughput but more realistic experiments. Therefore, given the circumstances, we adopted pre-training and fine-tuning training strategy. First, we pre-train a model for the target sequence-based prediction with the integrated target dataset, which we call it PTD, as in pre-trained deep CPF1. Then we fine-tune PTD into deep CPF1 with the endogenous target dataset to integrate the CA information. For the development of PTD, we use a convolutional neural network, which has shown great performances in genomics. The input sequence is converted to numerical matrix with the one-hot encoding. The convolution and average pooling layer learn to detect important motifs. The following fully connected layers learn more complex representations and perform integrative classification and regression. Intuitively, we can understand CNNs as learning position matrices to identify regulatory motifs which are hard-coded in the traditional approaches. The structure of PTD is shown in the middle. The numbers in the parentheses are the dimensions of intermediate data. So the PTD uses the composition layer uses 80 filters of size 5. The average pooling layer uses size 2. It has 3 fully connected layers of 80, 40, and 40 hidden units, and the finer output layer. For fine-tuning of PTD into DeepShift Python, we integrate CA information as follows. First, we take out the output of the last fully connected layer, which is a 30-dimensional vector. Here we represent with 10-dimensional. Then we add additional chromatin. Then we add additional fully connected layer with the 40 hidden units to modify and match the dimensions of chromatin accessibility to the output. Then we perform element-wise multiplication of the 2 vectors. Then use the output in the original regression output layer. This is called the CA integration layer, which integrates the high-level sequence representations with the CA. During fine-tuning, we only update the CA integration layer and the output layer so that the model could learn to incorporate CA information while avoiding overfitting. The final model is DeepShift Python. Next, I will show our experimental results. As I said earlier, there are 2 types of data sets. For the former, we have 4 in-house data sets, and for the latter, we have 3 in-house data sets and 3 more data sets from other independent laboratories. The number of samples of each data set are in the plot. So for pre-training, we use HT1-1 of 15,000 samples. And for fine-tuning, we use HEK-lengthy dataset with 148 samples. Next, I will show the experimental results. First, I will show the ranking performance comparison with the in-house datasets. Figure A shows the test results for the 3 integrated target datasets. The ones on the leftmost ones are the results from the proposed method. So comparing the conventional methods such as boosted regression tree and regularized linear regression, which were the state-of-the-art at that time, the PTD outperformed them with a small but statistically significant amount. Then in figure B, we show the test results for the 2 endogenous target datasets. Again, the leftmost ones are the results from the proposed models. From each set, the left ones represent the results of the models, results of the fine-tuned models, and the right ones are the results of the pre-trained models. So for all the models, the performances still show that large improvements by considering the CA information. And especially DeepShift Python showed substantially great, better performance compared to the other models. Next, I will show the classification performance of DeepShift Python. Figure C shows the test target sequences from the 2 endogenous target datasets divided into 3 groups according to the DeepShift Python schools. So the CRISPR activities show significantly large differences among the 3 groups. For the last, I will show the ranking performance comparison with the independent left datasets. So figure D shows the results for the 6 endogenous datasets, and the leftmost one is the result of the DeepShift Python. So for overall, the DeepShift Python shows great generalization performance. However, for some independent left dataset, they show lower correlations. However, since the data sizes are small, having only 22, 18, and 10 samples, the performance differences are just caused by 1 or 2 ranking misses, and we're gonna say that the results are not highly significant. So for I will conclude the presentation. So in this work, we tackle the problem of CRISPR-CPF1 activity determination problem. In other words, which target will become more easily with CRISPR-CPF1. So we proposed a deep learning framework called DeepCPF1 based on a convolutional neural network for modeling the target sequences, and has a multimodal architecture for considering the both genetic and epigenetic factors. To deal with the available data sets, we adopted pre-training and fine-tuning strategy and showed the significant performance boost in activity prediction in terms of Spearman's rank correlation. So for more information, please come find me at the poster session tonight. It's the BE665, or you can read the full paper as well. I'd like to appreciate my lab members and collaborators. So this concludes my presentation, and thank you so much for listening.
Thank you very much. That was a very clear presentation. Do we have any questions? Yes. Well, you guys, there are microphones here.
This is a simple question. I might have missed it. So what was your output you were training against? Was it a thresholded good efficiency of cutting versus not good efficiency of cutting, or was it the efficiency as a number per se?
Yeah, the efficiency as a number per se. Yeah, it's a regression problem. We are solving a regression problem, but we can use the score as a classification performance as well. Thank you.
Excellent talk. Over here. Did you happen to have time yet to look at off-target sites and whether this method is applicable to those as well?
For now, we are using it for just on-target prediction, but we are also interested in the off-target prediction as well. Actually, there was recently one paper was published in the Nature Biomedical Engineering, which was done by John Doench and Jennifer Iskarden, and they haven't used the deep learning algorithm, I think, but I think that's the interesting paper currently.
Thank you.
Thank you.
Thank you. Let's just thank the speaker again.
Thank you, guys.